outer membrane, inner membrane, cristae, and the matrix. Alright, so 6-carbon glucose molecule is about to be oxidized, meaning it's going to lose energy, lose its electrons to start. But to start this, an inactive enzyme gets activated by dephosphorylating 2 ATP into 2 ADP plus 2 PI, which is inorganic phosphate. The enzyme then splits glucose into 3-carbon sugars. Oops, didn't mean to write the 3. Called pyruvate. Whenever a bond is broken, electrons are released. These electrons are used to reduce 2 NAD and 2H into 2 NADH, and eventually go to cristae where the ETC chain is. But because it's such a long trip, there's a secondary enzymatic reduction. The NADH is further reduced to 2 FADH, which also heads to the cristae. But meanwhile, in a second active site, the enzyme phosphorylates 4 ADP plus 4 PI into the 4 ATP. And since we spent 2 ATP earlier, that leaves us with a net gain of 2 ATP. Now, if there is no oxygen is present, it's called anaerobic respiration. In animals, it makes lactic acid. In bacteria, it makes ethanol through fermentation. But if oxygen is available, we do aerobic respiration. The 2 pyruvate molecules enter the outer membrane and move into the matrix of the mitochondria. Inside the matrix, we have 2 3-carbon pyruvate molecules. An enzyme called coenzyme A begins an oxidation reaction, breaking off 1 carbon from each pyruvate. These carbon combine with oxygen to form 2CO2, which is 2 carbon dioxide, and electrons from the broken bonds are used to reduce 2 NAD and 2H ions into 2 NADH, which are sent to the ETC ETC. And now coenzyme A has another side job, and it binds to the remaining 2 carbon acetyl groups, forming acetyl-CoA, which stabilizes the molecule so it can safely enter the Krebs cycle without breaking down. Okay, in the Krebs cycle, coenzyme A detaches and returns to be reused in pyruvate oxidation. Now the acetyl group breaks and releases and it joins with a 4-carbon molecule already in cycle. This triggers a long enzymatic process breakdown called dissociation, where bonds are broken and electrons are released. The 2 acetyl groups release their remaining carbons, which combine with O2 to make 4 CO2 total. And at each step, electrons are released and used in redox reaction. So 4 NAD plus 4 H ions are reduced into 4 NADH, and 2 ADP plus 2 PI are phosphorylated into 2 ATP, and 2 FAD plus 4 H ions are reduced into 2 FADH. The 2 ADP plus 2 PI are phosphorylated into 2 ATP, and finally 2 more, so NAD and 2H are reduced into 2 NADH. Those NADH molecules are now headed to the cristae for the next step. Okay, now we're in ETC. So there are 5 enzymes embedded here. So there's E1, E2, E3, E4, and E5, and the ATP synthase. Okay, NADH is oxidized at E1, releasing NADH, which is recycled back earlier stages. The freed electrons move up. So it's here. It moves up through the enzyme chain, gaining energy. The energy is used to pump H ions from the matrix to the intermembrane space, building a high concentration gradient at E5 or at ATP synthase. Those H ions need to return to the matrix, but they can only do so by flowing through the ATP synthase, and this is called reverse chemiosmosis. This flow activates the enzymes, then phosphorylates 3-ADP plus 3-PI into ATP molecules, which meanwhile O2 is the final electron acceptor. It combines with 2 H ions and electrons to form H2O water.